Now that we've discussed the physiological requirements of Clostridium botulinum, we can move on to discuss its one and only virulence factor, a very degradative enzyme called botulinum exotoxin. This exotoxin is the most powerful naturally occurring toxin on the planet. To put its toxicity into perspective, it would only take 39 grams of pure botulinum to wipe out all of humanity. So, to understand the function of botulinum exotoxin, it's important to understand its structure. Clostridium botulinum produces seven antigenically distinguishable varieties of botulinum labeled A through G, but only A, B, E, and F are responsible for botulism disease seen in humans and other vertebrates. So that's all we're going to be concerned with in terms of virulence. Botulinum neurotoxins are secreted as large proteins that consist of a light chain, seen here, and a heavy chain up here, and the two chains are linked by this disulfide bond. The heavy chain is responsible for binding the toxin to an axon terminal, and the light chain lyses proteins involved in the exocytosis of acetylcholine. We'll talk more about this process later. Once cleaved from the heavy chain at this cleavage site here, the light fragment becomes the most potent toxin found in nature. So when environmental conditions are favorable, Clostridium botulinum spores germinate, and it is this germination that initiates the secretion of botulinum exotoxin. Clostridium botulinum and the associated exotoxins can enter the body via the bloodstream by way of a breach in the skin in the case of wound botulism, or through intestinal absorption if the spores are ingested, and these spores are resistant to the acidic stomach acid. It's important to note that the presence of Clostridium botulinum is not necessary for the disease botulism to take place. Only the exotoxin is required. Once inside the bloodstream, the botulinum toxin accesses neuromuscular junctions via the capillaries. So, to recap, this whole process, this whole mechanism of action can occur separately from the bacteria. So the bacteria itself can germinate and produce the toxin in one environment, and the toxin can somehow access the organism through a separate mechanism, and the toxin will still be ingested and either enter the bloodstream or go through the intestinal barrier, disseminate through the whole body, and bind to axon terminals. So this can all happen without the bacteria itself being present. So to understand the disruption botulinum toxin causes, it's important to know how neurons operate under homeostatic conditions. Healthy muscles contract as a result of a process called excitation-contraction coupling, which begins with electrical signals traveling from one neuron down the dendrites, across the synapse, into the next neuron as a result of an electrical cascade resulting from the depolarization of sodium ions. So, from the spinal cord, this signal eventually reaches the neuromuscular junction, which is this little space between the final axon terminal and the muscle cell itself. From here, snare proteins, these guys right here, catalyze the exocytosis of acetylcholine containing vesicles from the presynaptic axon terminal. These vesicles travel across the neuromuscular junction and bind to acetylcholine receptors on the surface of the muscle cell. And this is what initiates depolarization of sodium-gated ion channels, initiating the excitation or contraction of the muscle. Botulinum toxin is a protease, which is a type of enzyme that breaks the peptide bonds of proteins. In the case of Clostridium botulinum, it denatures these snare proteins that are needed to exocytose acetylcholine. As you can imagine, without acetylcholine crossing the neuromuscular junction and activating muscle fibers, the contractions cannot occur, and this results in paralysis. A paralyzed organism cannot contract any of its skeletal muscles, and those include extra respiratory muscles that are inhibited because they function the same way. So the affected organism eventually asphyxiates because it can no longer breathe. So this raises the question of why this occurs. What benefit does the bacteria confer from essentially paralyzing and killing its host? Well, the current hypothesis is that by paralyzing and killing their host, Clostridium botulinum creates anoxic conditions for itself, since the organism's cadaver is no longer breathing and undergoing aerobic respiration. So the resulting necrotic abscess, or the dead body tissue, is an ideal environment for Clostridium botulinum because, as we discussed in the last video, it meets its temperature, acidity, and most importantly, its oxygen requirements for survival.